Yes, so I'll start now. So hello and welcome to our Clinical Genomics webinar this month. I'm Mariam, the Science Communication and Marketing Officer at GenomeArc. Today I will be hosting our guest, Dr. Meraj Khan. Dr. Khan is a clinical research scientist working in the Hospital for Sick Children in Canada, Sick Kids. He has also founded his own startup company for health solutions through artificial intelligence, Digibiomics. Today, he will be giving us a diverse view of neutrophil extracellular traps and their association with inflammatory diseases, from clinical research to large-scale big data analytics. It's a very rich topic covering diverse fields, and we're very excited to learn more about it today. Thank you very much for joining us today, Dr. Khan.
Yeah, thank you, Mariam. And I'm really thankful for you and for the GenomeArc for giving this opportunity and platform to so that I can share my work and definitely I will be also sharing what the progress my team is making in terms of data analytics. So let me quickly share my screen and then we'll walk through.
Sure.
So thanks again. And today's topic is to discuss about the neutrophils extracellular traps, nets, and a journey from clinical research to big data analytics, how we started using neutrophils and what we found and what mechanism we understand and what is our next step. So let me quickly go to the agenda, what I will be covering today in this webinar. Definitely, I'll go through the background and I will touch what clinical research we have done, what we found and we are doing, and from where we pivot from clinical research to big data analytics so that we can deliver or we can do some translation research. And I will be shifting towards data analytics and AI. Whenever we said big data analytics, then there is a need of AI, artificial intelligence, and I will cover that part. And I will finish up or end up my talk after sharing some projects which are in progress. So we all know neutrophils. Neutrophils is an innate immune cell, and neutrophils acts as a first line of immune defense. So whenever there's infection, inflammation, or pathogen attack, neutrophils comes first, and they recognize the situation. And we know that neutrophils have two critical um, um, characteristics, like they phagocytose. So when neutrophils see the bacteria or pathogens, and they eat it and they kill it. The other is degranulation. When neutrophils infiltrate, they release certain enzymes that destroy the surroundings. And these are the two key steps. I'm not going to go in detail because this is well-known aspects. But what happened in 2004, there is a German scientist, Arturo, and his a big group. They discovered something else. And what that leads is the discovery of neutrophil extracellular traps or nets, and the whole process is called a netosis, formation of nets. In that case, what happened, and let's say a situation where pathogens are in millions. I mean, the pathogens are more, but neutrophils are limited. So in that case, neutrophil is not going to eat one by one and not going to apply the phagocytosis phenomenon. Uh, so what then the situation, how the neutrophils is going to handle that situation? In that case, neutrophils die itself and releases their DNA inside out and form a net-like structure. And that net-like structure has lots of antimicrobial peptides. So those structure will trap and kill the pathogens. I'm going to show the detail about that. So before that, so this is the video I'm going to play, and that video shows, and actually we did this experiment in our lab. What we did, we took a neutrophils and added bacteria, Staphylococcus aureus, and we used two dye, the blue dye, which is a DAPI, that stain the nucleus DNA, and DAPI is permeable to cells. It will go directly to the cells inside the nucleus and stain it. While the other dye, which is in green, you can see here, is a cytox green dye that also stain nucleus, the chromatin, but it's not permeable to neutrophils, to the cells. So what happened? We can only see the green when neutrophils is exposed or neutrophil cell membrane is captured. So now you can see this video. So see here, as the time permits, the bacteria interacts with the neutrophils and they made neutrophils to start making nets. Some of and then you can see the dye which is blue, then now it's turned to green. So this is the process we recorded in our lab when we performing experiment to understand the nets. So as I explained in the previous one, and this diagram, this illustration shows what are the key steps in making nets. So whenever there is an agonist, pathogens, LPS, or ionophores, and PMA is a pharmacological agent. They interact with neutrophils. Neutrophils started decondensation. The detailed mechanism will be coming to the next will be covering to the next slides. And then the chromatin, the chromatin which is polymorpho, I mean, diff shape, it now is decondensing, and after some time, within 2 to 3 hours, now that it starts releasing. Why it starts releasing? What are the reasons they start condensation? Decondensation, sorry. This that we can cover in the later part. And this is the electron microscopy images taken in our lab. See how the Pseudomonas aeruginosa is trapped in nets, and the below panel is the confocal microscopy of neutrophils healthy, and the agonist PMA and ionophore, how they make the nets, and we can stop these nets by using some drug which inhibits this process. That I will cover in drug screening part. So this study was very exciting initially, and every scientist in this field, they were excited. Wow, we found a phenomenon that helps in combating pathogens or infections, and that was quite relevant. But as we progress in understanding this phenomenon and we studied, and there are more research uh, uh, combined and they share their outcome, it is also reported that it is not always good. What does it mean? Let's say there is a situation, neutrophils came in, infection happened, and the neutrophils eat up all the thing by releasing nets. But if these nets are not cleared, one thing. The second thing, if these nets are too much, or maybe neutrophils, there is some dysregulation of in the system in the microenvironment, neutrophils started making too many nets, too much nets, then that will be deleterious to the surrounding of that tissue, and that cause lots of problems. And as I'm gonna explain, that it starts from lung infection, like in cystic fibrosis, even in COPD and asthma, causes the exacerbation of situation in autoimmune diseases, and even it helps in cancer metastasis. There are reports where neutrophil nets, which are in blood circulation, they in that case the metastasis or the cells, they stick and they spread from one location to other, and it helps in metastasis. So that's the side effect, or the other effect the scientific community found. And because of that, everyone was everyone means every researcher started looking what is the mechanism, how we understand what what are the factors which regulate neutrophils to make nets or not. And if we understand the mechanism, then definitely we can find out or screen some drug that helps to regulate the neutrophils. So when we need the nets, we can use those drugs to make neutrophils to make more nets, or when we don't need, then we can regulate neutrophils not to make excess nets. So that is our purpose, and because of that, me and my whole team, we started looking deep into the mechanism of neutrophil extracellular trap. So, and this is as a slide which is kind of shows very how these nets are clinically relevant, and in what situation nets are good and what situation nets are bad. And there are some situations we don't know, maybe good or bad. So it's a huge, there is a huge potential to explore the mechanism, and then understand, uh, 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 and then find out the targets for drug screening or drug development. So if you can see here, in case of wound healing, this is nets are good because if there is a wound, so neutrophils make nets and they kind of cover up the wound so that further infection is confined, or the infection is not able to spread. So in this situation, nets are good. But in case of the cystic fibrosis, so what happened in the case of cystic fibrosis, the neutrophils are not, they are dying because of nets process, because, and then that chromatin or the DNA along with antimicrobial peptides, they kind of clog the, they kind of make mucus more viscous, and because of the more viscosity of the mucus, patient, especially the kids, they are unable to breathe. And so initially that phenomenon was not known. Every scientist or clinician, they know that maybe this is because of the just DNA of any cells. No, it is from it is the DNA or chromatin of the neutrophils in nets. And excuse me. And for that, what happened? The clinician started giving the DNAs to clean up the airway, and though that helped, but now we know, and there are foolproof evidences that nets kind of exacerbate the CF situation. And we all know that in COVID pandemic, net was the target that was hit by coronavirus. And in that case, there are publications and studies, especially in case of the severe late stage patients. There are the consolidation bodies, and those bodies were tested, and we found that those bodies are not only the debris of cells, rather they contain major amount of nets. So nets were also involved in that situation, and because of this consolidation bodies are the debris, are exactly the major component of the nets. They kind of causes the pneumonia, they causes the ARDS, and leads the destruction of lung, and that leads to the many secondary issues. So nets are good in terms of combating the infection and inflammation up to a certain extent. But if there's a dysregulation in the process, in the signaling or the mechanism of formation of nets or netosis, or the nets are not cleared from a local environment, that is not good. That causes many disease exacerbations, as I mentioned. So understanding of nets mechanism is important, because that will help us to see the role of nets, because role of nets is a kind of double-edged sword. We don't know whether it's going to be good or bad, but that role will help us to, the mechanism will help us to define the role and how we can use these nets for the purpose of treatment, or for the betterment of patient conditions. So as I mentioned, so we did we applied many approaches to understand this mechanism, and the one approach which I'm going to share here is the transcriptomics. So what I did here, we took the neutrophils, treated with the two different agents. One is PMA, one is ino4. Both are the agents which start signaling cascades to make nets in neutrophils, and the control. And we performed the transcriptomics analysis of all these neutrophils from three different conditions by using Affymetrix, and the Affymetrix have around 100,000 gene transcripts pro. And when I did the exploratory analysis to see how the overall data is coming along, so we found the three discrete clusters are the principal component, and when we label it, we found that these clusters are totally separate, and that makes sense because the clusters of controls, no treatment, PMA and ino4, they all have the separate component, and that tells the quality of our samples. Along with that, we went to see the transcription is the profile of transcription on each chromosome level in three conditions. And we can see here, in case of ino4, the transcription is happening too much compared to PMA, and that we know that when we perform the netosis kinetics, the ino4 treated neutrophils, they releases their DNA very fast. So means their kinetics is much faster, and in between 120 minutes or 2 hours, that the neutrophils are done to make nets if you use the ino4. And here PMA, the netosis kinetics is slow, but what happened actually, it's a kind of detailed discussion and sharing more data. But what happened in PMA? PMA is a kind of that mimics the path of infections, while inophore it's a kind of sterile injury. So in both situations, neutrophils response is different. One in infection situation or in injury situations. And we perform again the networking and analysis to see what are the transcription factor, because here in the neutrophil in nets the main component is chromatin. So something happened in chromatin, that's what chromatin started decondensation and then lead to the formation of net. So we studied the role of transcription factors and the different kinases. So we found a set of transcription factor which activated in PMA mediated netosis, the different set in inophore that I will summarize in this diagram, and this flow chart I will be coming to the next slide in a zoom in mode. So based on the previous large scale transcriptomic studies and some other studies, what we formulated this mechanism. And in this mechanism, we identified that nets or netosis happen in two discrete pathways: NOx dependent netosis and NOx independent netosis. As name suggests, here the NOx ROS reactive oxygen species is involved, while here in the independent, the NOx ROS is not, but mitochondrial ROS and calcium influx is involved. And in both pathways, their starting point is different. The involvement of kinases like ERK, JNK, AKTP38, SRC, they are different and some are common as mentioned here, but ultimately they attacked or they start decondensation of chromatin. And you can see here the decondensation starts, and there's an enzyme called as Gas-Dermin D that helps to make pores, and from the pores, the chromatin comes out and make a nets. But in case of this NOx independent, there is another factor, it's called PAD4 enzyme. PAD4 enzyme, what happened because of the calcium influx, this enzyme activates, and after activation of PAD4, this activated PAD4 comes into the nucleus and it does the citrullination of histone. What does it mean? It converts the arginine positive charge into citrulline, and because of the citrullination, the charge dynamics changes, and that decondenses more faster. And that's the reason, if I can go back here, we have more transcription firing in inophore mediated, because that pathway is much faster compared to the PMA, and that also involved the citronation of histone. But finally, the nets are same, and in that case, we have found these are the different targets: ROS, kinases, citronation, and histone, and even we have checked the pH and salt of the environment that also contribute, or that also plays an instrumental role in making nets or not. So with this information, with this mechanism, definitely we want to translate it. And for the translation, I did manually actually a drug screening. That was a tedious task, but I was fascinated to find out some molecules to see whether they inhibit netosis or not. And I borrowed drugs from an MD Anderson Cancer Research Institute, around 126 drug, and did the manual screening. And I found there is a class, it's called as anthracycline. And in anthracycline, there are 4 drugs: idarubicin, epirubicins, and 2 more. They inhibit the netosis, as you can see here, in all the conditions. And we can see here, so these are the beautiful structure of nets that stain with DNA, citrullinate histone, as I mentioned in the previous slide, and MPO is myeloperoxidase, is also a key in neutrophil enzyme. So we can see here how the nets, the strings are there. Along with that, these enzymes are these target proteins, citrullinate histone and MPO are there. But if in case of the drug, the netosis is completely inhibited. So coming to the other interesting part, so let's divert from too much molecular biology or basic science research to the data analytics. This is the era of data. We say data as a gold here. So the purpose is, there's a lots of data available in medical sciences and even non-medical sciences. So I'm going to focus more in towards healthcare medical sciences, but data analysis are gaining the sourceful information from a particular data is not easy. It's a very complex process, as you can see here. The first thing is the access. When we access the data, the quality of the data, who is accessing, what are the biasness in that data, along with that, what kind of technology we are using to get the good data, and what is our strategy to make uh, uh, planning or the execution so that we can get benefit or we can get a good, we can gain the biology out of it, and definitely the ownership and resources. So it's a huge complex process. There are many more factors which I have not listed or maybe I'm not aware of, but from in but our purpose is how we can automate the whole process so that we can start capturing the data from different resources, whether it's clinical images, whether it's histopathology, scans, clinical data, genetic data, or genomic data, and make a and get the information from there seamless, and all the information we can homogenize and start from different location and to get one consolidated outcome. So this is our approach here, and this is something which is needed for any sorts of studies related to healthcare. In the advancement of data analytics and AI is lagging in healthcare compared to the other field. Is this is because of the availability of the data access, the privacy concern, ethical concern, so many things. So that is something our team is working with this initiative to make the data available for the purpose of a particular requirement. Let's say there is a in GenomeArc, there is a need of getting genomics data for a rare disease. So how we can process the whole end-to-end pipeline so that there is a seamless availability of that data so that they can do further research. So just here, I'm going to show a glimpse of the importance of AI in healthcare, and and that will help us to understand how much AI is important. AI means data analytics and use of technology to find the recommendations or the information. Because of there's a need of AI because of the aging populations. We have recently gone through the pandemics. There is a problem of rare diseases. Costs are kind of rising, and we have a very limited workforce in healthcare systems, starting from A to Z, all the way, whether they are clinicians, other practitioners, or researchers, there are limited workforces. And there is a forecast of around 9.9 million medical staff shortfall up to upcoming 6 4 7 years. So there's a huge opportunity for that, and we can leverage AI and data analytics for giving a virtual assistance, giving personalized treatment. We can also help leverage the potentials of AI in screening, early detection, so that we do not have pandemics, we do not have outbreaks. If we can early screen and control it, definitely this will reduce the cost. Definitely this will help us to synthesize hypothesis and then work on it, rather keep working, doing experimentation, and then getting a hypothesis and then work on it. So this will help us to help even the researcher to innovate or to directly work on objectives or hypotheses, rather exploring the hypothesis. And definitely there would be challenges because of any path that is going to beneficial. There will be challenges. The challenges would be the data collections. The variability in the human data is a lot. Not only the ethnicity, not only the geographical situation, even within that, how and who and what is the process of collecting the data. Data collected in the morning is different compared to the afternoon and in the evening. So that's a huge challenge, and definitely the data scientists, data engineers are working in that domain. And the explainability of the data, what data we have collected, whether are we going to explain it, are we going to leverage it to gain the biology out of it. That is another challenge, is because we just don't want to collect the data, and then at the end we are not able to explain it. And the last two components are actually is our human nature, whether the human because this AI and the outcome is directly impacting or related to humans touch, whether they are able to change this management, change this or not, whether they are going to accept it. So we have to build that trust and we have to educate them, because nowadays, I mean, mostly what happened early or even currently, any patient they wants to have a clinician or physician touch. That is something which we always prefer, but differently down the line, because of the based by giving more educations, change management, we will be able to make our community trust on these AI based help and assistance and recommendations. So to conclude this slide, definitely we can deploy the AI, and AI can be deployed by in combination with technology to build new capabilities and help assist in healthcare. So I will be kind of covering some ongoing projects. They are of diverse domain, covering healthcare, screening, and some understanding the disease mechanism, and along with that, how we're going to collect the data. So, I mean, bear with me. Initially I started from neutrophils to all the way to the mechanism, drug screening, and now I'm kind of diversifying. So, and the purpose of diversification is to see how we can leverage and in what ways we can help the healthcare. So this is our project. It's called a therapeutic. So we are working here in terms of combating, screening, and giving precise medication of depression patient. This is a challenging, I mean, it's a problem, not only in North America, even in the entire globe. Every third or fourth people are having some sort of mental issues. And in that case, the antidepressants, they are not working. The antidepressants working only 30%, or we can say in a different way, the patient for any antidepressant, they only respond 30%, means 70% patient have to come back again after 4 weeks, and they get the different combination of antidepressant. This is a huge burden. And in that case, what we want to use AI. So we want to not use only the clinical history and clinical data of the AI, the depression patient, because that could be a subjective assessment. We want to add the EEG data, and further, we want to collect the genomic and genetic data along with the environment to predict the patient's the severity of depression, along with that what kind of antidepressant that particular patient need, so that he do not have to come back again and saying that this time that antidepressant didn't work, he has to come back for the next time. So this is a uh, pilot project. We have a very enthusiastic early data and findings, but we want to integrate more data component, not only the EEG, not only the clinical history, rather to get some genomic data, and that will really help us to define and find the precise medication. And definitely here, I may come back to GenomeArc and get help in terms of the genomic aspect, because that is not my core expertise. So because I worked in lung infection, and mostly the neutrophil and netosis part, I dealt with the lung, cystic fibrosis, COPD, and asthma. So I am interested how we can leverage AI and imaging to diagnose the lung problems. And there is a severe problem of lung related to pneumonitis and idiopathic pulmonary fibrosis. What happened in this case of HP and IPF, their symptoms and their image features are so close that most of the time it misdiagnosed. So we want to leverage here the technology. We want to leverage here the machine learning algorithm, so that we can train those characterized images and by in our ML models, and those then our models can help to diagnose and differentiate between both HP and IPF. So in that case, we still working, we collecting data, and hopefully we'll have some result very soon. The the again the here the interesting part is we do have lots of images related to H&E histopathology images of head and neck cancers. So we developed a model by training images with metastasis and non-metastasis, because here as I mentioned, nets are also associated in cancer metastasis. So my part was to associate with that. So in the early objectives, we kind of differentiated the in between. We have the differentiation in between metastasis and non-metastasis of head and neck cancer by using the H&E images. But now we are adding more data related to the genomic profiling of those patients. Along with that, the clinical reports of neutrophils and nets, so that we can move to the next step, not only just recognizing whether these images are indicating of metastasis or not, or what stages, rather what kind of signaling pathways and what kind of genetic profiling is there, so that in future we can kind of give a screening kind of information, whether a particular particular genomic profiling kind of or particular medical clinical information may lead toward this kind of disease or not. So this could be our focus is not to diagnose or screen based on the only the H&E, because that could be a very late approach, but early by integrating more data sets. So this is the other a big aspect which we are trying to do, because for any AI related projects, we need data, and the data is something is a challenging aspect. So what we want to develop a kind of a data bank, like blood banks available. In that data bank, we going to collect the data related to not only disease, rather collect data based on anatomical locations, the chest data, abdomen data, heart data, I mean, brain data. And along with that, we will be also collecting the genomic data, because that is related to many, even many disease or many disorders, as including rare diseases. So that when we have our data bank will build strengthen. So what we can do, we can do then the disease specific studies, or we can share this data to other institute. Let's say our company or our any institute is trying to work in related to breast cancers, and we have the data, so they don't have to wait for years and years to collect the data. We can share the data, so that process can be much faster, and the whole community will be benefited. So this is our approach to collect the data, all sorts of the clinical data, uh, so that we can have we can share and grow towards AI deployment and implementation. So coming to the last project, which we recently submitted to a grant, and that is very interesting, and that actually combines most of the things. So this project is focusing to an autosomal recessive polycystic kidney disease that is called as ARPKD. So this is a rare disease, and in that case, what happened, the after the birth, there are so many complications related to kidney. That is a more clinical part. I'm not going to go through. So what we propose here, so we're going to take uh, do the whole exome sequencing analysis or target sequencing analysis, and based on the sequencing data, so we will have that genetic um, map. Along with that, we're going to collect the clinical data, the environmental data, the geo-specific data, and combine everything. So that the whole the data set from 3-4 component on, not only the genomic, it will help us to find the and find the pattern in those patients geolocations, clinical history, and even the family screening, to kind of screen the whole population. And in that case, what happened, this will also help us to kind of give us early recommendations or early warning in those situations, so that before even birth of that baby which is having ARPKD, we can provide some clinical intervention, and that patient or that baby can be better managed clinically. So this is our whole big project, and this we submitted in Saudi Arabia in one of the institutes. So along with the expert who have expertise in ARPKD, and my team will be supporting in the domain of AI and data analytics. And definitely we may need a GenomeArc support towards the genomic things, and that is kind of my next next big project which we will be going to deal with. So I know I have to keep in mind the timing because there are many more projects I listed here, but I may not go to all. So I think I already took around 35 36 minutes. So I want to take a break here. If there's anything, Mariam, I can come back and I can share more details, but this is what I thought of. I prepared for this webinar for 30 to 35 minutes webinar. And please feel free to have any questions or comment suggestions. And this is my team in Secrets and my mentors, and many of them are working with my startup company DigiBiomics. So I'm thankful, and I cannot forget to recognize and acknowledge them. And for any further questions, suggestions, please feel free to reach me out through email or LinkedIn or phone. So again, I want to thanks GenomeArc, Mariam, and Dr. Mahmoudouddin for this opportunity to share what we are doing and in which direction we are going. And thanks again for this time and consideration. Thank you, Mariam.
Thank you very much, Dr. Khan. It was a very interesting topic. It was very rich and diverse. You really included a lot of perspectives. So I had a couple of questions. So in terms of the nets, the neutrophil extracellular traps, how do you think genomic studies can contribute to our understanding of how we can implement precision medicine in the case of inflammatory diseases that are caused by nets, in the bad case scenarios?
Yeah, so that's actually a very good question. So we cannot directly do the genomics of nets because neutrophils is already fully differentiated cells, and making nets or not, this is an instant phenomenon or characteristics. So, and this is not a genetically directed. So neutrophils decide whether they have to make nets or not based on the environment, right? But so there's one part. The second part is, and that is the good aspect of neutrophils. We leverage neutrophils to combat the pathogen, the infection. The bad side is neutrophil is associated with many disorder where it exacerbate the situation, exacerbate the clinical problems. Let's say in autoimmune diseases, in cystic fibrosis. Cystic fibrosis is happening through a particular mutations. There is a huge study, and there's lots of genomic studies has been done, and mutations has been recorded, and they are well known. So neutrophil or nets is not the cause, but neutrophils exacerbate the problem. So genomic studies will be relevant and needed as per the disease situation, which disease we are dealing. Let's say ARPKD. ARPKD is the autosomal disorder, and there is a mutation, and neutrophils comes in and they make more nets and they exacerbate the situation. So if we want to take this disease to understand and define and find the treatment, then we need the genomic things. So genomic profiling or sequencing or understanding, we need it in a combination with the nets understanding. And the third thing, we need it based on the disease situations. The third thing, I want to give an example. We recently worked on a proposal in the breast cancer. So in the breast cancer thing, we studied the mammogram. We have the data of histopath. We have the data of immune cells, and we have the data of the genomic things. What kind of genome profiling is? We want to combine everything, and then based on that, our data bank or models are now trained in a way, they are trained sufficiently so that they can tell any, and in that situation, any data we will get, our model will help to predict, oh, this could be problem A or B. So that is our holistic approach. And in that case, genomic profiling and genomic information is important, very important. Even in mental health, I mentioned initially people say mental health is not related to genetics. Nowadays, there are evidences, not only in many problem like ADHD, schizophrenia. There are relevance related and correlation of genomics. So we want to expand as a holistic, not we want to define in one aspect.
Yes, so
Yeah.
it's really disease dependent and can shift based on that. So genomics contributes, you mean, in part, but there are different aspects as well that we need to look at.
Yeah, exactly. That is true. And that could be based on the disease types and kind and what approach we're going to do that.
Yes. So another thing in the data analysis, you've described multiple complexities in the analysis process, and the potential benefits in integrating AI in different parts of healthcare and in different parts of analysis. So in your opinion, what is the most part that can really get improved by artificial intelligence? Like you've shown different projects, they're all very impressive in imaging and in different aspects. But what do you think is the most important thing that can get improved with AI integration?
So, I mean, I consider the key aspect here. The data is there, and data is there in every location. The data is sporadic. Like, let's say a hospital in UAE has their own data set, and the way they reserve or they store, they have their own ways of labeling and all sorts of things and then storing the data. Is the hospitals or healthcare in Canada, they have their own way. So the challenge is it- and and we cannot build any tool which by leveraging one data set from one region, right? That will be biased. So if you want to integrate, the homogenization is a tough part. The way data collected, the features, the labeling, and all, even in the machine and the instrument is different from one location to other and the third. So how we can homogenize it? So there are that in that case, the AI is the one. AI means machine learning and the end-to-end pipeline that helps. And in that and for that there is a process. There should be a universal global data dictionary things. So based on that data dictionary, if we follow, then our data storage will be in one format, anonymized, and any in future this can be integrated very easily, very quickly. So that is my approach, and that's why I am trying to work on making data banks.
Yes.
So they will follow a same guidelines of data dictionaries, and in those data dictionaries, every information will be there. And let's say if you want to homogenize data from Canada to UAE, we can share the data dictionaries, and then we'll quickly homogenize it. And in that case, high computational power and ML algorithms are needed, and that's what I'm hoping. And that is a key development in this era
Yes.
of AI.
I completely agree. It's very important to have like a standardized process that we can use in different regions and with different populations.
Yes.
The topic was very interesting, Dr. Khan. Thank you so much for joining us today.
Thank you. Thanks for your time and consideration. Thank you very much.
Thank you.
Bye.